Let's start our lecture with a hypothetical experiment. In this experiment, we will take cell A and assume the resting membrane potential of this cell at minus 60 millivolt. Now, we will stimulate this cell with a stimulator. Stimulator is nothing but a source of current, but with some controlled things. It is the positive terminal of the stimulator and it's the negative terminal. We will put the positive electrode towards the outside of the cell and negative inside the cell. And to capture the response, we will put a voltmeter. Voltmeter has a positive terminal and negative terminal. So as we know, the membrane potential is by convention inside value minus the outside value. So based on this, what should be our positions of the electrodes of the voltmeter? So positive electrode or positive electrode will be placed inside and negative electrode will be placed outside. Then only we can get the value of inside minus outside. So this is our setup for the stimulation and capturing the response of the cell membrane. Let's say we will give some stimulus. We will give an impulse stimulus. Let's say this is a impulse stimulus. This is the strength of the current and this is the duration for which I have given the stimulus. Let's say stimulus we give in picoamperes and we will see the response of the cell. So as we know, cell is at minus 60 millivolt resting membrane potential. Once we give the stimulus, the membrane potential raises to some value and again when stimulus is off, it again comes back to some resting membrane potential. So this is a response of the cell when we will give this sort of stimulus or impulse stimulus. Now we will increase the strength of the stimulus. Let's say we give this much stimulus, we have increased the strength of the current, but we will take the time same. So in this case, the response is something like this. The membrane potential also goes to some higher value, and again, when stimulus got cut, it again come back exponentially to the resting membrane potential. So in this way, we can see how stimulus is given and the response we get. Now we will do the same thing with cell B. Let's say this is a stimulator with positive end and negative end. We will stimulate using the electrodes and we will capture the response with a voltmeter. Negative electrode placed inside and positive placed inside and negative outside. Now in this case, we have given the same stimulus. Now the response of this cell, we will take its resting membrane potential again at minus 60 millivolt. So once it is at, there is no stimulus, cell is at minus 60 millivolt. Once stimulus is there, the membrane potential rises to some value and again it comes to the resting membrane potential once the stimulus got off. Now we will increase the strength of the stimulus again. This time stimulus is something like this. So let's see what happened to the response. This time again potential goes to some value, it depolarizes and after some threshold, we call threshold, it fires like this and it again come back to the original position, even it goes beyond the resting membrane potential and after some time it again come back to the original resting membrane potential value. So what we have seen in the response of the two cells. In this cell, we have observed as we increase the stimulus, the response also increases in a graded way. We will call this type of response as graded potential or graded response. This is known as graded response. We can see such type of response over dendrites and the receptors. This type of response is seen in muscle fibers or neurons. And when a cell possesses such type of response, we will call that cell as an electrically excitable cell, and the property we can call the excitability. This is the property possessed by these cells. So in this video, we will talk about the cardiac muscles. Cardiac muscles also possess this property of excitability, and the response we get, we will call the cardiac potential. So we will learn more about cardiac potential in this lecture. We will cover our topic in these following headings. We will learn about its phases, types of cardiac potentials and why they are required. We also learn how cardiac potential govern the heart rate, properties of cardiac muscle cells, and we will also learn about the ionic mechanism behind all these properties, and lastly the propagation of cardiac potential. We will not go in sequence, but we will try to grasp the concept. So first of all, these are the phases of the fast cardiac potential. So you should understand, whenever I say the word cardiac potential, always remember it is a response. It is a type of response. Okay. So response means if I have a graph between voltage, membrane potential and time, so this is the type of response that is exhibited by the cardiac cells. So this is one type of response. We call this response as fast cardiac potential. So first of all, we will go through the phases of this cardiac potential. So this phase is called the phase 0, and this is basically the depolarizing phase. We also call it upstroke phase. Upstroke phase. You should remember the names, or numbering is also very important. You should just not remember the name, but also the numbering of the phase. That is, phase 1 is known as rapid repolarization phase. This is the phase 2. Phase 2 is known as the plateau phase. And phase 3, when membrane potential come back to its resting membrane value, this is the phase 3, also known as repolarization phase. And lastly, this phase that is quite flat and near the resting membrane potential, this is known as electrical diastolic phase. So give some time to remember all these phases, because these are important. There are 5 phases: 0, 1, 2, 3 and 4. Remember these phases. Now we have a different type of cardiac potential, that is known as slow cardiac potential. इस cardiac potential में we have phase 0, phase 3 and phase 4. We don't have the phase 1 and phase 2. और जो इसकी phases है, इनको भी आप ध्यान से देखिए. You can find out the differences between fast cardiac potential and slow cardiac potential. फेज 4 जो है, पहले वाले में फ्लैट थी, इसमें थोड़ा सा अपराइजिंग है. और हम देख सकते हैं कि जो फेज 4 है, इस फेज 4 को हम बोलते हैं इलेक्ट्रिक डायस्टोलिक फेज. और इस केस में इसका जो पोटेंशियल है, इसको हम बोलेंगे डायस्टोलिक पोटेंशियल और पेसमेकर पोटेंशियल. और जो फेज 4 का जो लोएस्ट पोटेंशियल है, इसको हम कहते हैं मैक्सिमम डायस्टोलिक पोटेंशियल. नाउ वी विल फोकस on the differences between the fast cardiac potential and slow cardiac potential. You just see, this is a slow cardiac potential and this is a fast cardiac potential. And also remember here, this type of cardiac potential occurs in SA nodal cells and AV nodal cells, and fast cardiac potential occurs in atrial fibers, Purkinje fibers and ventricular fibers. Now as far as phase 0 is concerned, here the phase 0 and this phase 0. So one difference between these is, this is very steep. Okay, यहाँ पे जो slope है वो इतना ज़्यादा steep नहीं है. Okay, तो हम यहाँ कह सकते हैं slope less steep, slope is very steep. फर्स्ट फेज और सेकेंड फेज तो यहाँ प्रेजेंट ही नहीं है, तो वी विल क्रॉस इट. इट इज प्रेजेंट हियर. नाउ अबाउट द फेज 3. फेज 3 के अंदर अगर हम डिफरेंसेस देखें, तो एक डिफरेंस तो ये नजर आता है कि इसकी जो अगेन स्लोप जो है वो स्टीप नहीं है, और इसकी स्लोप जो है स्टीप है. तो फर्स्ट थिंग, अगर स्लोप की बात करें, not steep. Here slope is steep. Now, one more difference is here phase 3 follows 0, phase 0. Here phase 3 follow phase 2. Okay. Follow phase 0. Here follow phase 2. अभी जो differences हम देख रहे हैं, after a time you will learn why these differences are important and what is the effect of these differences. जैसा कि हम देखेंगे यहाँ पे जो phase 3 है, it may control the heart rate. वहाँ पे जो phase 3 है, it is controlling the relative refractory period and other things like relaxation of the muscles and other things. Now come to the phase 4th. Here phase 4th is uprising. Here phase 4th is flat. Phase 4th is uprising. Here phase 4 is flat. Even we can say जो फास्ट कार्डिक पोटेंशियल्स हैं यहाँ पे वी कैन से अ रेस्टिंग मोमेंट पोटेंशियल सॉर्ट ऑफ थिंग. यहाँ पे एक रेस्टिंग मोमेंट पोटेंशियल होता है जो कि -90 मिली वोल्ट रहता है. और इस केस में एक्चुअली रेस्टिंग मोमेंट बिकॉज दिस ये जो कार्डिक पोटेंशियल्स है यहाँ पे हम देखेंगे कि दीज सेल्स हैव द प्रॉपर्टी ऑफ ऑटोरिथमिसिटी. तो ये स्टेबल रहता ही नहीं है, रेस्टिंग कभी स्टेज पे रहता ही नहीं है. तो वी कांट कॉल इट रेस्टिंग मोमेंट पोटेंशियल, बट हाँ हम कह सकते हैं कि ये जो लोएस्ट पोटेंशियल है यहाँ का दैट इज अबाउट -60 मिली वोल्ट. और इस लोएस्ट पोटेंशियल को वी ऑलरेडी नो वी कॉल इट द मैक्सिमम डायस्टोलिक पोटेंशियल. नाउ वी हैव सम, यहाँ पर भी ये भी एक डिफरेंस है कि जो फोर्थ फेज का रेस्टिंग मोमेंट पोटेंशियल है आरएमपी जो है यहाँ पे, या जो लोएस्ट पोटेंशियल है दैट इज -60 मिली वोल्ट. यहाँ पे जो है रेस्टिंग मोमेंट पोटेंशियल जो है दैट इज -90 मिली वोल्ट. So these are the differences. Now the question is why there are two types of cardiac potentials, and the answer lies here. As we know, every cell has got a specialized function. Some cells in heart are specialized to do the function of impulse generation. Few cells are specialized in impulse conduction, and few cells are specialized in contraction. So based on their function, they have got different type of cardiac potentials. If we say there are 5 types of cells in heart, one is the acinodal cells, another is the atrial fibers and AV nodal cells in the atria, and Purkinje fibers and ventricular fibers in the ventricles. So you can see the acinodal cells and AV nodal cells have similar type of cardiac potential. So these both type of cells are more important for the impulse generation. These cells, atrial fibers and ventricular cells, are their main function is contraction, and Purkinje fibers, their main function is conduction. Gradually we will learn how the different phases of cardiac potentials are customized to these functions. In the previous slide, we have learned that every cell has got some special function, and to fulfill that function, that cell has some special properties. So in this slide, we will focus on the cells having slow cardiac potential. And these cells are the SA nodal cells and AV nodal cells, and we will try to find out some more properties these cells have. So first, I will draw a slow cardiac potential. In this graph, this is a graph between voltage and time, and this orange line, we will take it as the maximum diastolic potential, and this green line, we will call it threshold. Now I will draw a graph of slow cardiac potential. This is the phase 4th. Once the potential comes at threshold, it fires and it will fire like this. So this is phase 4th, this is phase 0, and this is the phase 3. Again, it will goes to the threshold. You can see the slope of the phase 4 here and here is the same. So in this way, it will fire, again come here, and again it will fire, come here, and in this way this goes on. So what you are seeing here, slow cardiac potentials auto-generate themselves, means they have the property of auto-rhythmicity. So one new property, that is auto-rhythmicity. So first property we learned is the excitability. Another property of the cells who possess slow cardiac potentials is the auto-rhythmicity. Now we will see the effect of a change in the slope of the phase fourth. ठीक है कि phase fourth जो है इसकी slope अगर change करते हैं तो क्या effect आता है, and this will lead to a property known as chronotropy. Now I will draw one more diagram of slow cardiac potentials with a slope that is decreased in phase 4. So let's say this is phase 4 and its slope is decreased. You can see the slope of the red line, it is decreased, and again once it comes to threshold it will fire, sorry, again comes here, and again this is the slope. You can see the slope here and here is same, and it will fire another action potential. So what you are seeing here in the blue graph, we will call it control 1, we will call it control 1. In the red graph we decrease the slope of phase 4. So उससे क्या हुआ कि जो heart rate है, you can see we have got only two cardiac potentials here in the same time. So it means heart rate decreased. So what is the effect of, if I decreased slope of phase 4, effect is decreased in heart rate and vice versa. Okay, so this type of effect is known as chronotropy, means ability to change the heart rate. Now moving ahead, and now we will discuss the effect of shift of negative shift of the maximum diastolic potential. As we have already told, our maximum diastolic potential in the control we have taken this, and this is the threshold. Now if we shift the maximum diastolic potential on this side, this is a new potential, we will see its effect. So first of all I will draw a control graph. Control graph is like this only. Now I will draw a graph whose maximum diastolic potential goes down. So I will start from here. Now I will take the slope similar as this one. So slope is the same. The slope of the phase 4 is same as the blue graph. Now here comes the threshold. This will fire, again comes down. Now again the slope is same. So what you see here, कि if maximum diastolic potential shifts down, so again heart rate decreases. So what is the effect of is decrease in the heart rate. So in this way you can see how cardiac potential is affecting the heart rate. Taking one more example, in this example we will change the threshold. Let's say this is the maximum diastolic potential and this is the threshold, and this is a new threshold. First I will draw a control graph. This is a control graph. Now I will draw a new cardiac potential with having threshold at a higher value. I will draw in red. Again here thus we will take the slope and maximum diastolic potential same. So because threshold has increased, so this potential has to come here before fire. So here this is the slope 0 and this is the slope 4. So you can see the slope of red line here and here is same, and here it fires and goes down, again it fires like this. So what you are seeing again, the heart rate decreases here also. So what is the effect of threshold? Once threshold is at higher value, its effect is decrease in the heart rate. So what we have seen, there are many factors like the slope of the phase fourth, shift of the maximum diastolic potential, and the threshold, shift of threshold. These all things affects the heart rate. And this property of change in the heart rate, ability to change in the heart rate is known as chronotropy, is known as chronotropy. So till now we have learned about three properties. One is excitability, another is auto-rhythmicity, and third one is the chronotropy. One new property we will going to discuss is about the conduction velocity, ability to change the conduction velocity or impulse conduction. In this slide, I will try to explain the effect of slope of the phase 1 on the conduction velocity. Let's say we have two cells, Purkinje fibers, cell A and cell B. Both have their resting membrane potential at minus 90 millivolt. Okay. Now, somehow cell A got excited and fires a action potential that is known as the fast cardiac potential. So I will draw here. Let's say this is minus 90 millivolt and its cardiac potential will be like this. So we can say at some instant its potential becomes 10 millivolt. अगर 10 millivolt potential यहाँ है और यहाँ ये वाला जो सेल है, सेल B is still at RMP minus 90 millivolt, तो एक पॉजिटिव पोटेंशियल से नेगेटिव पोटेंशियल की तरफ एक करंट फ्लो होगा और ये जो करंट फ्लो होगा ये भी इस पोटेंशियल को क्या करेगा, थोड़ा ड्रॉप कर इसका पोटेंशियल जो है बढ़ाएगा. ओके. अभी ये जो फायर हुआ था, स्टिल एट दिस टाइम ये जो बी सेल है, दिस इज अ, ये सेल ए सेल का है, दिस इज द फॉर सेल ए. आई विल ड्रॉ द एक्शन पोटेंशियल फॉर सेल बी. अब आप देख सकते हैं कि जो सेल बी है, ये इस टाइम तक तो दोनों ही रेस्टिंग मोमेंट पोटेंशियल पे थे. ये फायर हुआ तो इसका थ्रेशोल्ड मान लिया कि ये वाला है, दिस इज द थ्रेशोल्ड. ठीक है, जैसे ही थ्रेशोल्ड पार किया तो ये ऊपर चढ़ा, ये इसने फायर कर दिया. बट ये वाला जो सेल है, जब ये सेल इस पोटेंशियल पे होगा तो हो सकता है कि क्योंकि जब करंट उसके पास आ रहा है तो इसका भी थ्रेशोल्ड बढ़ना स्टार्ट होगा. तो इसका थ्रेशोल्ड जब बढ़े, अभी स्टिल इट इज एट अ, इसका थ्रेशोल्ड बढ़ने लगेगा, धीरे 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 बढ़ने लगेगा. एंड सपोज आफ्टर सम टाइम, इसने तो यहाँ फायर कर दिया था, इसने आफ्टर सम टाइम इट फायर्स. सो व्हाट इज गोइंग टू हैपन, अगर इसकी स्लोप फास्ट हो जाती है, इसकी स्लोप अगर स्टीप हो जाती है, इफ इट इज स्लोप गॉट स्टीप, तो व्हाट विल हैपन, ये जो है जल्दी थ्रेशोल्ड पर पहुंच जाएगा एंड इट विल फायर अर्ली. ठीक है. सो द व्हाट वी हैव कंक्लूडेड हियर कि अगर जो स्लोप है, स्लोप ऑफ फेज वन, स्टीपनेस है तो कंडक्शन वेलोसिटी increases, and this property is known as dromotropy. This property or ability to change in conduction velocity is known as dromotropy. In this slide we will see the effects of long duration of the phase 2 of the cardiac potential. In muscle fibers, skeletal muscle fibers, we know electrical activity is followed by the contractile activity. But in case of skeletal muscle fibers, the duration of electrical activity or action potential is very less. However, the duration of contraction is quite large. So we can get a phenomena of summation there, or we can call it tetany. Now, in case of cardiac muscles, this is not happening because the duration of action potential or cardiac potential is about the same as the duration of contraction. We will also learn that the absolute refractory period of the cardiac potential is also governed by the phase 2, and we will see these phases reflect themselves on the ECG as QT interval. To conclude, we will revise what we have learned in this lecture. We have learned few properties like the chronotropy, that is the ability to change the heart rate. Bathmotropy means change in the excitability of the cardiac fibers by changing the threshold. Auto-rhythmicity, this is present in the slow cardiac potentials because of the phase fourth or pacemaker potentials. We have also seen the ability to change the conduction velocity. The conduction velocity is governed by the phase 1 of the phase 0 of the cardiac potential. Similarly, there are other mechanical properties in which we have discussed that tetany is not present in the cardiac muscle fibers. However, there are other mechanical properties like inotropy, that is the ability to of contraction. The lusitropy is the ability of relaxation, and other biochemical properties like the non-fatigability. At last, we have some questions like what are responsible for the different phases of cardiac potentials, why the resting membrane potential of fast cardiac potential and slow cardiac potential are at different values, what governs it, why are phases 1 and 2 are absent in slow cardiac potentials, and why is phase 4 is different in both cardiac potentials. So there are a lot of questions we have to answer. All these questions will be answered once we have understanding of the ionic basis of the cardiac potential. So in the next video, we will talk about ionic basis of cardiac potential. Thank you. Thanks for watching.